This is the second video on the immune system, and in this video we're going to take a much closer look at the adaptive immune system. So the adaptive immune system is specific, meaning that it will respond to one very specific pathogen at a time. It takes a lot longer to respond than the inflammatory process does, and unlike the inflammatory process, it is systemic. It also carries a memory of pathogens that have been encountered in the past, and we'll see how that works later on in this presentation. So adaptive immunity has two components to it, humoral immunity and cellular immunity. Humoral immunity is basically substances that are flowing in the blood plasma. And so this means that humoral immunity is going to be antibody mediated. And so these antibodies are going to be produced by B lymphocytes or plasma cells. And antibodies are going to respond to bacteria, to bacterial toxins, and to free viruses in the bloodstream. Cellular immunity involves cell-mediated destruction of foreign cells or of chemicals that cause inflammation. Cellular immunity is going to be carried out by T lymphocytes, and they are mainly going to attack, and we've seen a similarity to this before, virally infected cells, parasite-infected cells, cancer cells, and cells of foreign grafts, in other words, donated organs. Now, I realize that the T lymphocytes sound very much like what you've heard about natural killer cells, but we're going to talk about the specific differences between these T lymphocytes and the natural killer cells later on in the presentation. So what are antigens? An antigen is a substance that can cause an immune response. Most of them are large and complex molecules that are not normally present in the body and molecules that are recognized by the body as non-self. So there are a couple of different types of antigen. A complete antigen, which has the ability on its own to activate lymphocytes and antibodies. And this ability to activate lymphocytes and antibodies is something that is called immunogenicity, literally meaning to start immunity. A complete antigen also has reactivity. In other words, it has the ability to react with activated lymphocytes and antibodies. And so basically, you have to be able to be bound by lymphocytes and antibodies for the immune system to work. And so that's the reason why a complete antigen not only has immunogenicity, in which it can activate lymphocytes and antibodies, but it also has to be able to be bound by these lymphocytes and antibodies. And so what is a complete antigen? Proteins are the strongest complete antigens. Nucleic acids can be complete antigens, and some lipids can be, and also large polysaccharides. Now, incomplete antigens are something that are different. So incomplete antigens involve smaller molecules that can only start an immune response when they get attached to one of the body's proteins or a large carrier such as a protein. And so incomplete antigens do tend to be small molecules such as peptides, nucleotides, and hormones. So how does this process actually work with an incomplete antigen or hapten? So basically, the reactive portion of the hapten, with the protein carrier, once it's bound to the protein carrier, it can then itself bind to antibodies. So when this hapten combines with the body protein, it can cause a full immune response. Okay, now this response can be harmful in the case of allergies. And so what are some common allergens? Penicillin, poison ivy, animal dander, detergents, and cosmetics. So actually, there's one thing that I learned when I had a pretty severe autoimmune reaction and I had to go to a rheumatologist. They did an allergy profile on me. And so what I found out is that a reaction to an animal dander is not really a reaction to the skin itself, to the skin flakes themselves, but a lot of animals, particularly cats, which I'm just deathly allergic to, cats clean themselves by licking themselves. And so it's not so much the skin of the cat as it is the remnants of the saliva that the cat has licked itself with. And so these haptens that are in the saliva are actually derived from blood plasma. So it's actually a part of the animal's blood that you're allergic to in many cases with animal dander. Okay, so the specific portion of a molecule that causes an immune response is called an antigenic determinant or an epitope. And as you can see here, most antigens have got multiple different antigenic determinants, multiple epitopes. And look at the top, both the top diagram and the bottom diagram show you three different things. It shows you three different shapes, and it's the actual shape of the antigen, which is what the antibodies respond to and bind to. So you can see three different shapes on the antigen above, three different shapes on the antigen below, and on the antigen below, it's actually being bound by three different antibodies. So it's actually a pretty terrific thing for us that most antigens have got multiple epitopes, because it allows us to respond to an antigen with multiple antibodies. And as a result, our response is that much more thorough. So with an antigenic determinant, each epitope causes a specific immune response. Now, the reason why I've got plastics mentioned here is because plastics are actually, most plastics are actually polymers. And so you see right here that you've got styrene here, and what's the polymer of that? Polystyrene. And so the thing that happens here is that the plastic themselves, the monomer of the plastic itself, doesn't cause a reaction. So if the monomer is not a reaction, then the polymer is not going to be a reaction. So again, remember that plastics are a simple molecule with repeating units. Okay, and because, again, the monomer doesn't cause an immune response, neither does the polymer. This is the reason why a lot of plastics can be used as artificial implants. Okay, so let's talk about antigens that are presented on the surface of cells. And I realize that we went over this in the prior video, but I wanted a refresher here because I'm going to make a bit of a comparison in the next couple of slides. So self-antigens are these MHC proteins that we talked about before. And MHC stands for major histocompatibility complex. And again, there's a combination that's unique to individuals that identifies the cell as being self or belonging to your own body. Okay? And you've got MHC1 molecules on all body cells except for red blood cells. And so that's particularly fortunate for us that there are no MHC1 molecules on red blood cells, because this means you can donate blood without worrying about tissue typing, the same type of typing that you would have to think about with, say, a liver transplant. And so the lack of an MHC1 molecule on red blood cells actually allows us to accept blood from individuals from whom we would not be able to accept something like a heart or a liver transplant. And so let's take a look again at the process of forming an MHC1 molecule. So you have an antigen that is produced on the inside of the cell. This antigen then gets digested into fragments. At the same time, you have MHC1 molecules being produced, and the fragment and MHC1 molecules get combined in a vesicle, and that vesicle gets transported to the cell surface. And what this results in is a cell being able to show cells of the immune system, yes, I'm a normal cell, yes, I'm producing normal proteins, no, I don't need to get killed. Now, again, the MHC proteins are taken to the cell surface. Now, if an intracellular pathogen, such as a virus, has hijacked protein synthesis, let's take a look at what happens here. So the virus infects, and the cell starts producing viral proteins in addition to the endogenous antigens. So you're going to have viral proteins that are bound to the MHC1 molecules, and as you can see, the viral proteins get packaged up and get displayed on the surface of the cell. And so what this really does is to prevent pathogens like viruses from hiding in cells as effectively as they might otherwise be able to, because as long as the virus is having the cell produce the viral proteins, those viral proteins get presented on the surface of the cells that are producing them. Okay, now the next thing that we're going to take a look at is a totally different type of MHC antigen. Okay, MHC still stands for major histocompatibility complex, but in this case, instead of grinding up and displaying a foreign protein, you're going to get, a self-produced protein. You're going to get this cell actually taking in an exogenous or an exterior protein. So it's going to phagocytize it. It's going to digest it into fragments. At the same time, it's producing MHC2 molecules. And as with MHC1 molecules, these peptide fragments get combined. They get combined into vesicles where they fuse. These vesicles then get migrated to the cell surface. So what these types of cells do, they're not displaying the work that they're doing. They're displaying what it is that they found. So you only find MHC2 proteins on the surfaces of some macrophages, some dendritic cells, and some B lymphocytes. These are specific cells that are called antigen-presenting cells. So they don't come up to cells of the other cells of the immune system and say, look what I'm producing. They come up to other cells of the immune system and say, look what I found. And I've had a student in the past call these NARC cells, and if that's the way you want to remember them, that's fine. They report what foreign proteins are being found in the bloodstream. So B lymphocytes are one of the cells of the adaptive immune response. And so B lymphocytes take a role in humoral or antibody-mediated response. So here's a picture of a B lymphocyte. T lymphocytes take part in a cell-to-cell action of the adaptive immune response, and here's a picture of a T lymphocyte. Now, as I just mentioned, you have antigen-presenting cells, which are macrophages, dendritic cells, and some B lymphocytes. Now, their job is to present a foreign protein to one of the other cells of the immune system, and we'll go through the mechanism of this in more detail in just a little bit. So all of these B and T lymphocytes originate in the red bone marrow. The T cells become immunocompetent in thymus gland, and we'll describe this process in just a little bit. So this is a 2 to 3 day process, and the cell division produces a lot of different lines of lymphocytes, but only about 2 of them actually become immunocompetent. The other 98% get screened out. So the way they get screened out is they're tested to see if they attack self, but they're also detected to see if they can detect non-self. So there are a couple of different standards here. Do you detect self but not attack it? If you attack self cells, you get screened out. Can you detect non-self and then attack it? If you detect a cell as non-self but you don't attack it, you're pretty much a useless lymphocyte and you get screened out in that case. So these two processes are called self-recognition, and the other process we'll cover in just a minute. So self-recognition is when T cells can recognize your own MHC proteins, and they basically have to do so by recognizing MHC proteins on thymic cortical epithelial cells, which I know that sounds like a mouthful, but the thymus has a cortex, and the cortex is lined with epithelial cells, and those epithelial cells display self-MHC proteins. And so the cells that recognize that MHC protein get selected for. So this is self-recognition. Can you recognize something as self? The other test is self-tolerance. So if you have a T cell that targets self-antigens, they get selected against. So a self-antigen is any molecule that gets recognized by self as your own body, but would register as foreign in somebody else's body. So this is basically one of the ways that tissue typing for donation works. You look for similarities between self-antigens, and the closer that they are structurally, the better the chance that the recipient's immune system either doesn't attack or has less of an attack on the transplanted organ. And so these T cells interact with a different set of cells, dendritic cells that are located at the junction of the cortex and the medulla of the thymus. Now, this is a negative selection, unlike the self-recognition where the cells that recognized were selected for. These are T cells that get selected against. Okay, so if you're a T cell, first of all, you better recognize. Oh, sorry, I couldn't resist that. But you had also better not target your own cells. So again, positive selection means those lymphocytes that can recognize are permitted to survive. The rest of them undergo apoptosis. This is called the MHC restriction process. With negative selection, if you bind too strongly to self-antigens and have too strong a reaction to them, you get eliminated. And again, this is self-tolerant. So taking another look at it, looking at the top diagram here, if you're positive and react weakly with the MHC, then these survivors are kept. So you have a restriction of the surviving cells to the cells that react positively. In the second scenario down, you've got cells that fail to recognize the self-MHC. So you see that there's no binding between the T cell and the thymic cell, and these get eliminated by apoptosis. And in the third instance here, it's binding a little bit too tightly to the MHC cell. It's reacting too vigorously, and so these cells also get selected against, and they're instructed to carry out apoptosis. And so the surviving cells not only can recognize the self-MHC molecules, but they also tolerate them. Okay, so B lymphocytes go through a similar screening process. It's a little less well understood, but the result pretty much is the same, that you get the deletion of lines that either react too strongly to the cells or don't recognize the cells. And so the process by which B cells get eliminated is something that's called receptor editing. I don't want to go into too much detail about that. That's a little beyond the scope of the course, but the bottom line is pretty much the same as the T lymphocytes. If you don't recognize self, or if you react too strongly to self, you get eliminated. So this slide shows you sort of the functional cycle of lymphocytes. So first of all, they're produced in the red bone marrow, whether they're T cells or whether they're B cells. Now, if you're a B cell, you stay in the bone marrow. If you're a T cell, you actually migrate to the thymus. Now, they go through the immunocompetence procedure that I talked about. So this is the self-tolerance and self-recognition screening. Once you've undergone those, then you are an immunocompetent lymphocyte. And after immunocompetence has been established, then these cells leave either the thymus if they're T cells or the bone marrow if they're B cells. Now, at this point, they're called naive immunocompetent cells. And naive immunocompetent cells means that they have the ability to recognize a foreign antigen, but they haven't yet encountered one. And so these populations of naive immunocompetent cells will seed the lymph nodes, the spleen, other lymphoid tissues wherever you find antigens. And so once they're there, if an antigen is encountered and they're activated, then they're going to be considered mature immunocompetent lymphocytes, and these will circulate continuously in the bloodstream. They'll also circulate continuously in the lymph and the lymphoid organs of the body. Okay, so antigen-presenting cells. Again, these have the MHC2 molecules. This is a different diagram that shows basically the same thing. These types of cells are macrophages, dendritic cells located in connective tissue, Langerhans cells in the skin, and activated B lymphocytes. And so, again, the biggest thing that I want you to remember is that this is a foreign antigen that is being taken in, cut apart, and presented on the surface of this cell. So they present these fragments to the lymphocytes, and the T helper lymphocyte is the usual recipient. So basically what you've got here is a T lymphocyte in yellow and the antigen presenting cell in blue, and the antigen presenting cell saying, hey, T helper lymphocyte, look what I found. Okay, so this is kind of a process by which some of these things happen. So here we've got a little picture of a B cell. The B cell has a receptor and there are antigens. Now, one thing that I want to make clear is that you have got a huge variety of receptors on a huge number of lines of B cells. So your immune system has the ability to respond to up to about 10 to the 12 different antigens over the course of your lifetime. So just about any foreign antigen that enters the bloodstream will be recognized by at least a line of antigen-presenting cells. And there will be at least a line of B cells and T cells that can be activated from that. So here what you're looking at is this yellow asterisk is the antigen, and no, I did not think about the fact that this looks like the Walmart symbol before I put it in. I just put it in. Green is the receptor, and the MHC2 receptor is actually in red. So the first thing that happens is that an immunocompetent but naive B lymphocyte is activated when it binds to a foreign antigen. So you see the antigen bound to the receptor. The next thing that happens is the antigen and receptor complex gets actually pulled into the cell, endocytosis. This antigen gets digested, so you see that the antigen's been broken apart, and you can also see on the red MHC2 receptor that is being presented on the MHC2 receptor. So here it'll present it to a type of cell called a helper T lymphocyte. And what the helper T lymphocyte is going to do is it's basically going to say, okay, great, you found a foreign antigen, now proliferate. And so you're going to have a lot of copies of that B lymphocyte being produced. It will mature into a plasma cell, each of these lymphocytes, and these lymphocytes are basically like little antibody factories. They're going to produce a whole lot of antibodies. So you're producing an antibody that is specific to the original antigen that you encountered. The other thing that the helper T lymphocyte does, even though it's not shown here, is that the helper T lymphocyte will actually activate cytotoxic T cells that are geared to react to the same antigen. Okay, so going over the humoral immune response, most of these B cells become plasma cells, and so what they're going to do is synthesize and secrete an antibody, and they'll die after 4 or 5 days, but this is your primary immune response. Now, from this primary immune response, not all of the B cells become plasma cells. Some of them become memory cells that are stored in places like lymph nodes, and they get triggered if you encounter the same antigen a second time, and this causes a secondary immune response. So we're going to take a look at the primary and secondary immune responses in a bit and compare them. Okay, so looking at this, you see a primary response in initial encounter with the antigen. So the antigen is like the little reddish-orange dots. And you can see that only one of these B lymphocytes has the ability to bind to that particular antigen. Now, the B lymphocytes that don't have receptors that will bind to that antigen, they're not activated. They remain completely inactive. So basically what you're going to do is this, you're going to limit this whole process strictly to the line of cells that can bind to this particular antigen. So these are going to proliferate to form a clone. So you're going to get mass division of that one specific B lymphocyte. So these B lymphocytes will then, most of them mature into plasma cells. But again, so those plasma cells secrete antibody molecules. But again, a few of these B lymphocytes get held back as memory cells. Now, when you encounter a second challenge by that same antigen, and this response, or this can be a secondary response that occurs years later, you're going to have a clone of cells that's identical to the original cell up at the top, but this time you're going to produce a whole lot more plasma cells, which produce a whole lot more antibodies, yet retain more memory cells. So the thing that I want to emphasize here is that your secondary response is much more dramatic than your primary response. So your secondary response is faster, so it happens in 2 to 3 days. You have a higher concentration of antibodies, so more antibodies get produced. The duration of production is longer, so you have weeks to months of this higher concentration of antibodies. That makes it much more effective. And in this case, you have a different structured antibody that's produced. So here what I want to show you is basically a graph of a primary response and a secondary response. And so what's the first thing that happens? You get your first exposure to a particular antigen. You produce antibodies to that antigen as part of your primary immune response. Okay? Now I want you to notice the numbers on the x-axis, which is days, and the numbers on the y-axis, and look at the numbers on the y-axis. It says 10 to the 0, 10 to the 1, 10 to the 2, 10 to the 3, 10 to the 4. Okay? So as you go up each one of these blocks, you're not talking about just adding one, you're talking about multiplying by a factor of 10. Okay? So by the time you go from, so a response at the third block produces literally 100 times more antibodies than the response at the first block. Okay? So take a look at the peak of our secondary immune response. It's way up there close to 10 to the 4th, so it produces almost 1,000 times as much antibodies as your primary immune response. So when I said it produces a lot more antibodies, it really does. Now look at the slope of the line too. The slope of the line goes from minimum to maximum a whole lot more quickly than it does with the primary immune response. And also take a look at the way that the curve tapers off. So with the primary immune response, you see that curve coming right back down. With a secondary immune response, which is up at the top, you see that curve reducing much more slowly. So you get a faster response, you get thousands of times more antibodies produced, and they're produced for a much longer time. So our secondary response to an antigen is much more dramatic and much more effective than a primary response. Now, there's a second antigen that's on this slide that's in sort of that rose color. So what this is intended to show you is that if you have this incredibly effective secondary immune response to antigen X already in place, if you're exposed to antigen Y, then your previous response to antigen X doesn't have a thing to do with your response. So every new antigen, every new pathogen requires a separate primary immune response. Okay, so what are the ways in which you can obtain active and passive humoral immunity? So first of all, what is active and passive humoral immunity? So we'll go over this over the next couple of slides. So active immunity is naturally acquired when you get the illness. Artificially acquired is when you get a vaccine for a particular illness. Now, vaccines only give you a portion of a particular antigen. They don't give you the original pathogen. Okay, so most vaccines involve inactive or very low activity viruses. And what this is going to do is either minimize the symptoms of the disease, or you shouldn't have any disease symptoms present at all. So basically what a vaccine does is to cause your primary immune response as if you had gotten the illness. So basically a vaccine is pretty much a fake illness that doesn't give you the actual disease. And so when you get the vaccine, you have your primary immune response, and that means when you encounter the pathogen later on, for the pathogen, it's its first encounter with you, but for your immune system, you give it that secondary immune response, which is way more effective. So the reason why you want to have this secondary immune response in place is because some pathogens are virulent enough to make you thoroughly miserable or kill you before your primary immune response can adapt to it and take care of it. And in a lot of cases, the disease can kill you before your primary immune system can really swing into gear. If you have a secondary immune response ready and waiting for this pathogen, then when you first encounter it, you're going to kill it. So this is the reason why immunizations are as effective as they are. Okay, so in passive humoral immunity, you get antibodies from another person or another animal. And memory, in this case, does not occur. And so you can get passive humoral immunity in a number of ways. One, by maternal transfer to the fetus. Second, by being given gamma globulins into your blood serum. And some examples of these will be snake antivenom and toxin for botulism, um, globulins for tetanus, for rabies, and for hepatitis B. Now, the animals that are used for manufacturing these antibodies include most commonly rabbits. I'm sorry. Um, these passive antibodies, they only last for several weeks. And see, the thing is about passive antibodies is that because your immune system hasn't done the production, you don't retain memory cells from this. And so if you run into the same problem again, you're going to have a second treatment with the passive antibodies. And so the animals that get used for manufacturing antibodies uh, most commonly include rabbits. And again, I'm sorry, these provide immediate protection. That's the good thing about it. Because think about this: if you get bitten by a snake, you don't want to wait several days for your primary immune response to kick in. You're going to be dead, and so it's better to have that antivenom in place. And so rabbits, chickens, goats, guinea pigs, hamsters, mice, rats. These are animals that produce antibodies, and the way that those antibodies get produced basically is that people inject those animals the particular with the particular antigens, and so those animals produce the antibodies. Those get isolated from the blood of the animals, and then that's where you get antivenom from. And so when you need particularly large quantities of antibodies, then you're going to produce sheep. And as you can tell, that sheep thinks that's a pretty bad proposition. You know I wouldn't be able to resist that. Okay, so of acquired immunity, you can have naturally acquired immunity and artificially acquired immunity. So the naturally acquired immunity can either be active, in which case you've had contact with a pathogen, or passive, in which case you've had antibodies passed from the mother to the fetus or an infant in her milk. With artificially acquired immunity, you can either have active artificially acquired immunity, in which you're going to produce the antibodies and produce memory cells, and so a vaccine is the best example of an artificially acquired active immunity. And you can have passive immunity, where you get injection of these antibodies directly into you. Okay, now the antibodies themselves, there's a basic antibody structure here. You've got 4 polypeptide chains. 2 of the chains are the heavy chains, which are in sort of that sort of purplish color, and 2 of the chains are the light chains, which are in sort of that bluish color. Now, the hinge region is where these chains are connected. Those yellow sort of dots are disulfide bonds. So these are sulfur-containing amino acids that are able to form covalent bonds between their sulfurs. And this is something that holds the structure of this antibody together very well. Okay, now in each of these, I want you to notice that there are things that say VL, VH, CH. Okay, CH stands for constant region of the heavy chain. VH stands for variable region of the heavy chain. So it's the variable region which differs one antibody from another. So if you have an antibody, say for chickenpox, if you have an antibody for influenza, the constant regions are going to be the same between those 2 antibodies, but the variable regions will either be tailored to fit the chickenpox or to fit the influenza. So basically, you've got constant regions and you've got variable regions. On each type in the variable regions, each antibody has got what's called a Fab region, and this is a site for binding the antigen. And then there's also what's called an FC region, which is not pictured on this diagram, which is where antibodies can bind to other cells of the immune system. I'm sorry, they actually are at the macrophage binding site. This would also be called the FC region. So not all antibodies look the same, but they've all got sort of that sort of Y-shaped theme. So you can see IgGs, these are the most abundant of antibodies, and so these will protect against bacteria and viruses. These are the only classes of antibodies that can cross the placenta from the mother to the fetus. Next one down is IgAs. You find this mainly in sweat, tears, saliva, mucus, breast milk, and GI secretions. And so um, these levels actually decrease during stress, and this is the reason why people who are under long-term stress are more liable to get sick. Okay, IgMs are the first antibody class to be secreted by plasma cells. So this basically, the IgM is the type of antibody that gets secreted during your primary immune response. It's the IgGs that are involved in your secondary immune response. So I think you can see how it would be easier to produce the IgGs than the IgMs. And why is it that the primary immune response involves IgMs and the secondary IgGs? I don't know, and I don't think anybody does. Okay, IgDs are found on the surfaces of B cells as antigen receptors. So these are the receptors that your antigen-presenting cells use to bind to antigens in the first place. Okay, IgEs get involved more in allergies than anything else, but they also can provide some protection against parasitic worms. Okay, so how is it that you get such a large diversity of antibodies? So it says on the third bullet point here that a human can make more than 10 to the 12th different antibody molecules. So the reason why is because you have a number of different types of segments: V segments, D segments, J segments, and constant region axons. And so basically you have, in the variable region, you have sort of this shuffling and recombination of gene segments, so that you might have one or two or three out of these close to 40 V segments. You might have between 1 and 23 of these D segments. You might have between 1 and 6 of these J segments. But the fact that all of these get recombined in different fashions give you an absolutely huge variety of structures on the Fab region of antibodies. Okay, now when it comes to cell-mediated immune responses, T cells are best suited for cell-to-cell interactions and killing. And so they're going to kill cells that contain bacteria inside them. TB is a good example. They're going to kill virally infected cells. They're going to kill cancer cells, and they're also going to kill cells from transplants. Okay, so T cells are what we see used in cell-mediated immune responses. So this is going to be cell-to-cell interactions and killing. And generally the targets that you're going to see T cells come after are going to be cells that are infected with some sort of internal parasite, like with tuberculosis or virally infected cells. They'll also target cancer cells and cells from transplants. So T lymphocytes, and you see the T lymphocyte in blue beneath here, they can only see and respond to antigens that have been processed. And by that, I mean antigens that have been presented by the antigen-presenting cells. So dendritic cells, and you're going to see one that's pictured here, that's sort of that cream-colored cell. Dendritic cells are one type of APC, so are macrophages, Langerhans cells, and again, some B lymphocytes. So basically the type of receptor that responds to a presentation to the APC is called a T cell receptor. So the first thing that I needed to talk about is the different types of T cells. So there are helper T cells, and helper T cells will actually activate B cells and activate cytotoxic T cells. So here's what happens. You've got millions of T cells. Each one of them has a unique T cell receptor, and this will bind to a specific foreign antigen. And what that means is you've got the ability to respond to several million types of foreign antigens. Now, if you look at the diagram, here you're going to see the T cell receptor on the cytotoxic T cell and the T cell receptor on the helper T cell. Now, along with this T cell receptor, you've got what's called a co-receptor, and these are the CD proteins. In cytotoxic T cells, it's going to be the CD8 protein, and in helper T cells, it's going to be the CD4 protein. So they look a little bit different, but their job is essentially the same. They help maintain the bond that's formed between the MHC2 and antigen and T cell receptor. So T cells also require an additional molecule for stimulation. Now that's not pictured here, but these are called co-stimulators, and one of the most commonly known co-stimulators is called interleukin-2. And so what interleukin-2 does is to not only activate your helper T cells, but they will also enhance the cell division, the proliferation of T cells, of B cells, and of natural killer cells. So once you get the activation of a T cell, it undergoes clonal selection. That's just a fancy way of saying that that particular T cell with that particular T cell receptor is the one that undergoes cell division. All of the other millions of other types of T cells, the ones with different antigens, they don't undergo cell division. So this is called clonal selection. Okay, now how the T cell responds depends on what type of T cell that it is, and I gave you a brief preview of the different types of T cells. So the T cells that display CD4 are helper T cells, and so these helper T cells actually secrete interleukin-2 that I just talked about, along with a variety of other cytokines. So this is their prime trigger of T cell proliferation, and here's what's going to happen. So this first helper T cell that encounters your antigen will produce IL-2, which will cause the cell division of that particular type of T cell, of that particular line of T cell. Those new helper T cells will themselves secrete IL-2, and so this is a positive feedback mechanism. So you'll see that you have a helper T cell get activated. It'll go through what's called clonal selection. So you'll have a lot of cell division and you'll form a helper T cell clone. And so what these active helper T cells will do is they'll stimulate cytotoxic T cells, and they'll also stimulate B cells into more rapid division and have them produce a lot more antibodies. And as usual, you're going to see some of them held back as memory cells. So basically the helper T cell helps the other cells of the immune system do their function. That's how that it actually gets its name. Now, T cells that display CD8 are cytotoxic T cells. So these are the cells that have sort of the direct action. So basically, here's what happens with these cells. So the helper T cells will activate the cytotoxic T cells. And so these cytotoxic T cells will carry out the same kind of thing, clonal selection, where they carry out a lot of cell division of that particular line of cytotoxic T cell, and then they go out for a search and destroy mission. They're going to attack infected body cells, or they might attack cancer cells if your immune system happens to recognize a particular cancer, or they may unfortunately be involved in organ rejection. And so most of the cytotoxic T cells that are produced actually go out, search and destroy, but some of them get saved as memory cytotoxic T cells, which are really long-lived cells. So what these cytotoxic T cells do, now the way that they carry out their action, and this is a T cell that's actually crawling over another cell, and it's preparing to destroy it. So they'll release perforin and granulysin, and so what perforin does is it's the same thing that it does in natural killer cells, punches holes in the target cell membrane, and granulysin can get in there and destroy intracellular microbes by punching holes in their membranes. So if you've got a tuberculosis bacterium hiding within a cell, the first thing that the T cell does is to punch holes in the infected cell and then punch holes in the microbes that are within the cell. Now, some cytotoxic T cells will release granzymes, and you've heard that before. These cause the target cell to undergo apoptosis. And so at that point, any microbes that get released or any cell fragments then get mopped up by phagocytes. Now, this is another thing that T cells do, and this is called immunologic surveillance. And so they'll stick to and crawl over the surface of other cells looking for antigens that they might recognize. And so I've often joked in class that T cells are kind of like the TSA of the immune system. So they'll crawl all over things and they'll feel up the other cells looking for a specific antigen that they can target. So sometimes this term immunologic surveillance gets reserved for natural killer cells and dendritic cells, but I think it's fairly applied to T cells because all three of these different types of cells will crawl over other cells looking for, in the case of T cells, either a specific antigen that it's targeting, or in the case of natural killers, anything but normal. So this is a summary of the different types of lymphocytes that are involved in the acquired immune system. So helper T lymphocytes, these initiate and oversee the immune response. And what they'll do is to activate cytotoxic T lymphocytes. They'll activate memory T lymphocytes. They'll activate B cells, which mature into plasma cells. And they'll also activate memory B lymphocytes. And so the helper T cells get everything started. So one of the things that actually makes HIV as bad as it is that HIV actually specifically hits helper T cells, and that cripples the rest of the immune system in terms of its reaction. So the T lymphocytes, the B lymphocytes, these are parts of the acquired immune response that responds to a single antigen, and the natural killer cell is part of the innate immune system that responds to multiple antigens and kills a wide variety of both infected and cancerous cells.